Hi, today I'm talking about adverse event prediction and drug repositioning through a relational graph convolutional neural network approach. My name is Sophia Krix and I'm a PhD student at the University of Bonn, and I'm also at Fraunhofer SCHI. In drug development, the majority of clinical trials fail at some stage, where hereby we face a two-component problem. We have a costly development process with high expenses and low success rates, and also adverse events occurring in late-stage clinical trials, which lead to a failure. We can tackle both of these problems as follows. Instead of starting the drug development from scratch, we can use previously approved drugs and reuse them for treating another condition. This is called drug repurposing. Since we want to avoid adverse events occurring in late-stage clinical trials in the first place, we can try to predict the occurrence of these in advance. We can achieve both of these goals with one method. We can do link prediction in a biomedical network. We represent part of biological knowledge as a graph with nodes and edges. And in this graph, we can then focus on specific edge types and predict if a drug could have a novel indication or if it could have an unforeseen adverse event. We can use specific convolutional networks that operate on graphs for doing link prediction. Considering the node VI, we gather information from neighboring nodes in the graph to make a single node update. This technique is called neighborhood aggregation, and it works as follows. Each node is represented by a feature vector. We gather information from the respective node's first order neighborhood by also taking the edge type into account. As we also want to integrate the node itself, we use a self loop for the node. These messages, they are all aggregated then to produce the updated node representation. Considering the graph input, we selected descriptive features for each node type with which we annotated the nodes. For the drugs, we used molecular fingerprints, gene expression signatures, and morphological profiles. For the proteins, we used sequence embeddings. And for the conditions, we made use of clinical concept embeddings. So how do we now integrate these multimodal features? We use a multimodal embedding layer in the beginning, which transforms multiple feature vectors into a 1D vector. This is then used as input for the RGCN, the Relational Graph Convolutional Neural Network. These architectures are forming the encoder. Now let's look at how we use this for link prediction. Considering a pair of drug and condition nodes, we want to predict whether a relation of type indication is likely to exist between these two. We use the multimodal input features for the encoder, which generates a latent embedding of both nodes. The decoder, a tensor factorization model, then generates a score for the edge between these two, taking the relation type into account. What we receive in the end is a probability score for the respective edge. And this works on one drug and condition pair, but this is what we can do on the entire graph. So now let's look at the results. We used our approach for the two use cases that I was talking about in the beginning, the adverse event prediction and drug repositioning, which are on the left and right respectively. So we compared our approach to state-of-the-art approaches for individual use cases. So on the left-hand side, if we focus on adverse event prediction, we compared it, for example, to the random forest. And for drug repositioning, we compared our approach to deep walk and shortest path. Overall, you can see that our model outperforms all of the other approaches. This here is an interesting example for drug repositioning predictions that our model made, which is suggesting to use Mifepristone for Alzheimer's disease. And indeed, this drug has been tested in several studies and was found to reduce the pathology of Alzheimer's, which is seen in the reduction of amyloid beta load and accumulations of phosphorylated tau. In summary, we developed an RTCN model which integrates multiple modalities. And our model also allowed us to accurately predict new indications for existing drugs and do drug repositioning, and then also to predict unwanted adverse events of compounds. These are my references, and thank you very much.